Here is the glycolysis. Most of you have seen this pathway. Either you're in ARP, coming from ARP, or in your bachelor's. This is the most common pathway that everybody sees. If you have taken biochemistry, that means you know glycolysis. If you say, no, I don't know, you are lying. It is being taught. The way it is taught, it is different, right? It may be boring at that time, but it is taught, right? What is glycolysis? Glycolysis is oxidation of glucose. That's it. You are oxidizing glucose. That is what is glycolysis. Your lysis. Lysis means what? You are breaking. You know, that's what is lysis. Lyse, break. What you are lysing, what you are breaking? You are breaking glucose. That's what is glycolysis. So there are certain enzymes which will help in oxidizing glucose. And what are those enzymes? How it is taking place? And that is what is shown in this figure. As you see there, look at the figure. Glucose is oxidized all the way to pyruvate. Okay. Now, as the glucose is oxidized into pyruvate, you see here, pyruvate in the bracket number 2. What is number 2? 2 pyruvate. 1 glucose breaks into 2 pyruvate. Okay. Now, in order to do that, in order to oxidize glucose into 2 pyruvate, initially your cell spends ATP because it is breaking, and it is spending ATP to initiate the process. And that's why, as you see here, there is ATP entering into the reaction and being oxidized into ADP. ATP enters, breaks the terminal phosphate, give energy, allow the reaction to go in forward direction, and then move on, right? And then again, as you see here, in the third reaction, there is another ATP entering in, and that energy is used to conduct the reaction in forward direction. Just to give you an overview of why ATPs are coming in, right? So just an overall general understanding, not an important thing, but you should know this. Why always ATP gets in? What is the purpose of this? So if you look at first reaction, conversion of glucose to glucose 6-phosphate, in fact, it is an endergonic reaction. You know endergonic reaction? Endergonic reactions are the reactions where you need to provide energy. They cannot happen spontaneously. You got to provide energy to do it. Like if I need to move this cup from here to here, that's endergonic. I have spent energy. And if your muscles are contracting, you are spending energy, right? How do you get energy? You have to provide external energy there. What is that extra energy? That is ATP. Whenever a reaction is endergonic, in order to make that reaction possible to go in a forward direction, you got to provide energy. And the energy currency for our cell is ATP. When you break a terminal phosphate in the ATP, convert that into ADP, you get minus 7.3 kilocalories of energy. And that energy is used to conduct the reaction or take the reaction in forward direction. And that minus 7.3 kilocalories, it is being used in other reactions as well. Like example, just to give you an idea, it's a bioenergetics. I don't want to go into too much into that, but you should know why these things are coming in, right? So, breakdown of this minus 7.3 kilocalories, that's what you get. Don't write it down. This is just a general understanding. You don't need to know the numbers. Okay, break ATP and you get minus 7.3 kilocalories of energy. And imagine that conversion of glucose to glucose 6-phosphate, and that needs energy which is plus 4.0. That much energy was needed. Okay, but how much you got? Minus 7.3 kilocalories. And later, the next reaction, glucose 6-phosphate to fructose 6-phosphate, energy that was needed was plus 0.5. And then fructose 6-phosphate into fructose 1,6-bisphosphate. Let's consider this reaction needs energy of plus 6.0 or something. Just a random numbers I'm writing. These are not accurate, but minus 7.3 kilocalories per ATP is accurate. Okay, let's consider like this. Now, you have $7.3, $7.30 with you, right? You go to Best Buy. All you have is $7.30. How much you can purchase from Best Buy? How much material you can purchase from there? Only $7.30, not more than that, right? If you purchase $7.50, you are 20 cents short. And the ladies there, they are very strict. St. Kitts ladies, I think they are really strict. Gents are good. You know, lady police. Anyone have vehicle? Drive your own vehicles, like rented? Be careful. You know, you should have your sticker and driving license, all that, because lady police, very strict. They're always keeping an eye, wearing seat belt, and immediately they give, $100, $150, sometimes 500 EC they give. Yeah, they don't even if you say sorry, they don't. They are very strict. Male police, it's very chilled. Okay, don't worry. Go. Go, man, go. So they do, but not these ladies. Anyway, I'm nothing against, but St. Kitts ladies are like police. That's what I have seen. Anyway, so they then 20 cents, they don't give discount. All you can purchase is $7.30. That's it. So here in this metabolic reaction, you have $7.30, and glucose to glucose 6-phosphate needs $4, right? So out of $7.30, you are going to spend $4. How much left? $3.30, right? And so as long as the next reaction needs $3.30 or less than that, you are fine. You can still use those $3.30. So next reaction, see, it needs only 50 cents. You have $3.30, so you can still conduct that reaction you're conducting. And then the next reaction needs $6, which you don't have right now. So what do? You use another dollar, you break ATP again. You break ATP and you get another minus 7.3 kilocals. So that minus 7.3 kilocals plus the previously remaining money. How much? Whatever it is, we didn't calculate. So that will add up. And then you move on. So as you move on, right? So down the line, there are some reactions which are in fact exergonic. That means they have so much energy in them that they are able to make ATPs. So this is what cell will do. Cell knows down the line, I'm going to gain. That's why it is investing. This is an investment. Cell is investing 2 ATPs initially, knowing that later I'm going to gain. It's like our general investments. Why you are investing? Why you invest? Anybody investing? You have worked and you invest money in real estate, stocks, mutual funds, whatever, gold, bonds. Investment is to gain later return. You're expecting a return. Cell also investing initially, it is going to invest 2 ATPs and later it is going to gain ATPs. So that's how the metabolic pathways works. Metabolic pathways always invest ATP to initiate the process and later it's going to gain more ATPs from it. So wherever there is endergonic reaction, ATPs gets in. Wherever there is a super exergonic reaction, ATP is produced. That's the concept. Okay. So now, with that understanding, let me take you into this. So there are 2 ATPs initially invested, and later you see in the glycolysis, there are 2 ATPs made in this reaction, 2 ATPs made in this reaction. That means 4 ATP minus 2 ATP initially invested, that will be net gain of 2 ATP. And that's what happens when glucose breaks into 2-pyruvate. So it is initially investing 2 ATPs. Later it's going to get 4 ATPs. All this is going on in the cytoplasm. Remember, generally where ATPs are synthesized, we have seen yesterday. Where is the location of ATP synthesis? That's in the electron transport chain, inner mitochondrial membrane. Remember, glycolysis is a cytoplasmic pathway. All these reactions are going on in the cytoplasm. And your cell is making ATP in the cytoplasm. Is it not strange? It's a strange thing. Your cell is able to produce ATP in the cytoplasm. See, there is ATP produced 2A plus 2 ATP down there. This type of ATP production in the cytoplasm without going into electron transport chain is called as substrate level phosphorylation. Means at the substrate level, outside of mitochondria, your cell is able to produce ATP. What is the electron transport chain referred as? What are the two other names for electron transport chain? Respiratory chain, correct, because it is related with respiration. And the other is oxidative phosphorylation. When you are oxidizing NADH, you are phosphorylating. That's oxidative phosphorylation. Whereas here, the substrate, when the substrate is converted to product, you are phosphorylating ADP with PI to make ATP. That is why it is called as substrate level phosphorylation. So there are two examples for substrate level phosphorylation here, and those are the two reactions. And that is what will be important. I'm going to show you later. Okay. So this is how oxidation of glucose is able to make a net ATP here. That's one thing. And also, as you see, there is generation of 2 NADH plus 2 H plus in the cytoplasm. And if your cell is able to transfer that NADH H plus into mitochondria somehow, so doesn't it contribute to ATP generation again? 1 NADH H plus is worth how many ATPs? Oh, what? No, ATPs. 10 is proton. ATPs. 2.5 ATPs. 2.5. There are 2 NADH H plus. That means 2.5 plus 2.5. 5 ATPs. Right? That 5 ATP plus these two down there. So 7 ATPs. So like this, ATPs can be generated in the cytoplasm itself and later in the mitochondria through those NADH plus. That's why oxidation of glucose is able to generate ATPs for the cell. That's one thing. Second thing is, there are these intermediates that you are getting here. They can be diverted into so many other metabolic processes so that other things can be produced. Right? So we are going to look into that aspect as well. So this is what in general how glycolysis happens, and the enzymes are mentioned on the side according to the numbers. Not all of them are important. So we are going to go into only the important reactions. Like there are how many enzymes? 10 enzymes are mentioned. There are a couple of them are not important there. So we are not going to go into those things. Okay. On this side, I have written a couple of things. One of the speciality of glycolysis is, this is the only pathway in our cell which can happen, which is able to oxidize glucose with or without oxygen. As I have written in this second statement, it provides ATPs to cells with or without oxygen. That's the speciality of glycolysis. No other pathway is able to do this. So glycolysis happens in the presence of oxygen or in the absence of oxygen. That means irrespective of availability of oxygen, glycolysis is able to provide ATPs to your cells. Okay. So aerobic oxidation and anaerobic oxidation, I have written aerobic condition, anaerobic condition. So under anaerobic condition, as I have written here, the product is 2 lactate. 1 glucose gives you 2 lactate. That's the basic information that you should know. In the absence of oxygen, what is the end product of glucose? If somebody asks, it is lactate, 2 lactate. If you don't answer that, it's not that they will blame you, me. Who is your teacher? Who is your professor? Dr. Mungli, what did he teach you? If you don't know that, no one says, but they think like that. Because it's a fundamental information. Glucose to lactate, that happens in anaerobic condition. Okay. Now, next thing is to know what are those anaerobic conditions. This is the important one. If you want, you can mark it down. 2 lactate, important. And then this one, super important. You got to know the conditions for anaerobic glycolysis. Wherever you come across these conditions, Your end product of glucose is lactate. That means you are referring to anaerobic glycolysis. And that's how the questions will be framed. So look for these scenarios. What are the conditions where anaerobic glycolysis is expected? Red blood cells. You know why? Because it doesn't have mitochondria. Red blood cells don't have mitochondria. It conducts anaerobic glycolysis. And then many cancer cells, preferentially they conduct anaerobic glycolysis. Not that they don't have mitochondria. They do. But preferentially they conduct anaerobic glycolysis. There is a phenomenon called Warburg effect that we are going to see. Even in the presence of oxygen and mitochondria, cancer cells, they conduct anaerobic glycolysis. That is called Warburg effect, Warburg phenomenon. So we are going to see that. Tissue hypoxia or anoxia, you know the meaning? Anoxia, no oxygen, hypoxia, less oxygen. So obviously anaerobic, prefer to go into anaerobic, such as myocardial infarction. You all know about, like, at least coming from anatomy, you know what it is, right? So coronary is blocked. So that area is not getting oxygen. So only that area which is not getting oxygen go into anaerobic. Okay. Other areas fine. And then skin graft. Why skin graft? Because when you take a thin slice of skin from maybe thigh and put it on to hand wherever there is a burn, and until that thin slice of skin gets its own new blood vessel, it has to survive by anaerobic glycolysis. Okay, so that is the another condition where anaerobic glycolysis is expected. And there is another condition, if you want, please write it down, called septicemia. Please add this, septicemia. Septicemia. What is septicemia? Septic shock. You heard of this? Septic shock. Somebody going into septic shock. Septic shock is, it's a bacterial infection. So much of bacterial infection that leads to vasospasm of your arteries. Imagine entire body, arteries go into vasospasm. Means they constrict and blood flow to the organs doesn't go on properly. So entire body goes into hypoxia. And that is what is called septic shock. It's a like total shutdown. You know, sometimes your laptops die. Is it not? We go through this. Laptop suddenly died. You don't know how. Any experienced this? Suddenly died. That's what happens during septic shock, just done. Right? And that is when anaerobic glycolysis is trying to take care of energy needs of the body. Right? So that is one of the example for anaerobic glycolysis that you should know. So make sure you memorize these things. Okay? So there are a couple of reversible, irreversible reactions in glycolysis. As you can see the arrows there. Look at the arrows. Pay attention to the arrows in that pathway. There are some arrows which are bidirectional. There are some arrows which are unidirectional. Okay. So there are three reactions with the unidirectional arrow in glycolysis. First, third, and the last one. Okay. So those enzymes are written here. Irreversible reactions in glycolysis, hexokinase glucokinase reaction, PFK1 reaction, that is phosphofructokinase number 1, and pyruvate kinase. I want you to know these three reactions, not for glycolysis, but for gluconeogenesis. I'm going to teach you gluconeogenesis later. In order to understand gluconeogenesis, you should know these three reactions. Why? Because gluconeogenesis is simply reversal of glycolysis. Gluconeogenesis, new glucose is synthesized. How about instead of running from top to bottom down like this, you conduct reaction in a reverse order. You take 2-pyruvate and go all the way up to glucose. That's what is gluconeogenesis. In order to do that, as you see, there are so many reactions which are reversible, right? There is so many reactions here which are reversible. Reversible in the sense you can use the same enzyme to reverse the reaction. This enzyme, it can go from top to down or down to top. Okay, same enzyme can conduct that reaction. It all depends on the flux of the reaction, concentration of the substrate and the product. So in either direction, this enzyme can conduct a reaction. So you don't need to worry about the reversible reactions in glycolysis when you are doing, reversible reactions, these reversible reactions when you are doing gluconeogenesis, because you simply use the same enzyme to reverse the reaction. But what you got to worry in gluconeogenesis is how to reverse an irreversible reaction in glycolysis. Is it not? That enzyme is only going from top to down, not from bottom to up. So those three enzymes which are irreversible, you got to bring new enzymes for that. Totally new enzyme to take care of it. And those are the enzymes that you will be remembering in gluconeogenesis. So as long as you know which are the three irreversible reactions that I should take care, you know gluconeogenesis. That's it. That is where this first point comes in. Not for glycolysis, for gluconeogenesis. Okay, next. Steps where ATPs are used, which I have already mentioned it to you. The second point, as you see, first and the third reaction where ATPs are being used. Those are the two enzymes. And then steps where ATPs are produced. So these are the two reactions where ATPs are produced. So I have written the name of the enzymes which we are going to go through individually, right? The names is phosphoglycerate kinase, pyruvate kinase. We are going to go through these reactions. It refers to substrate level phosphorylation. And then the most regulated enzyme in the pathway is PFK1, phosphofructokinase number 1. And that is this reaction here. That is the most regulated reaction in glycolysis. So these are the areas where, means couple of them I have written here. So let's go through the very first one. So most